was not there in the topic which was given. Pleiotropy and one more.
Wait. See, it's like all the topics that I have taught you within that would have been there. Getting my point?
I mean, the topics were inheritance in first gene, then second, and sex determination. There were many topics in inheritance of first gene, as well as many topics in inheritance of second gene. But then mutation and pleiotropy were completely different topics. It is not under any of these.
See, the portion was inheritance of one gene, two genes, and sex determination, right?
Yes.
So see, the whole entire thing is like I have given you bold headings because I'm supposed to give you the headings according to the topics that are there in the list. Okay, but it's like all the things that come in between, meaning like for example, from monohybrid to dihybrid, whatever things I've made you write in the class, those things were supposed to come. Getting my point? It's not a very clear-cut distinction. I had to give these topics according to the list that they have provided me with. Okay, but it's like all the things that I've taught you up until the last topic, that is sex determination, that would come. Getting my point? So in future also, if you ever get these kind of topics in your exam, like this topic, so just understand that up until the last topic that is mentioned, all the things that I've taught up until now that are there in your notes, they will come. Okay?
Thought was this particular topic because they mentioned it. These three topics.
I know, that's what I'm saying. It is according to the list that there is. But I have taught you several things in between that also.
Yes.
Right? So you should have studied all of it up until sex determination, because beyond that I haven't taught you till the time I made your paper. So I did till sex determination, meaning all the parts that I teach you. Getting my point? So in future also you pay attention to this thing, that even though if the particular topic that will be the last one in the list of the topics in your syllabus, it will be all the things taught up until that topic, because I couldn't mention it like that. That's why. Okay. All right. So today we are beginning with a new chapter, that is molecular basis of inheritance. Now for this chapter, there are some prerequisites. What are those prerequisites? I want your attention. Like if you had been paying attention 100% in my class up until now, I don't want 100%, I want 200%. Because this chapter is a lot about, it's like chemistry in your biology. Okay, so we gotta pay attention, and we're gonna see all the things that makes this chapter all sorts of the worst chapter in the world. Anyway, we'll not focus on that topic. See, in general, you know that the genetic material is of two types, DNA and RNA. And do you know what they are collectively known as? Like if I say DNA and RNA, what are they? In terms of biomolecules, what do you think DNA and RNA are? Do you remember I asked you to revise biomolecules?
Oh yeah.
That is.
But in class 7, we do not have any biomolecules.
How is that even possible? That chapter is there to haunt you all. It haunted me in my time, it will haunt you in your time. Huh?
Class 11. Yeah.
Biomolecules, yes. Anyway, see, DNA and RNA, they are collectively called as nucleic acids.
Oh yeah, yeah.
When I say to you, remember everything. They are called as nucleic acid, okay? And basically, they are polymers of nucleotide. What is polymers?
It is polymers are a chain. I mean, many monomers together and they combine to form.
Very good. Exactly. Polymers are the bigger molecules that are made up of several monomers, right? So, nucleic acid is a polymer of nucleotide. It means that these nucleotides are the monomers of nucleic acid.
Yeah,
right?
okay.
Now, see. So how is a nucleotide made? So first of all, do you know what is a now so you know what is a nitrogenous base, right? Nitrogenous
Yeah.
bases, they are of two types. What are the types?
Sorry, nitrogenous bases.
Are of two types. What are the types? So this is
I
the thing I taught you in the previous chapter also. Huh?
I know the answer, but then
You
it's
say
just
whatever it is in your mind, you say that. I'll see whether it is right or wrong. Say it.
so I know that nucleotide are made up of three things: nitrogenous base and all those. And nitrogenous base, guanine, this
Yeah.
adenine,
Yeah.
guanine, pyrimidine, and purines.
Yes.
Purines
Purines
of
and pyrimidines are the nitrogenous bases. Among the purines, adenine and guanine are there. Among the pyrimidines, thymine, cytosine, and uracil is there. Clear? So when
yes,
a nitrogenous base combines with a pentose sugar. Pentose sugar means a sugar that is made up of how many carbons? 5 carbons. So when these two things combine together, a structure that is made is called as a nucleoside. Okay? Now, you take the nucleoside and you add a phosphate group into it. Now it becomes a nucleotide. So all in all, you can say that a nucleotide is made up of nitrogenous base, pentose sugar, and phosphate group.
okay.
Can you quickly tell me the full forms of DNA and RNA?
Deoxyribonucleic acid, ribonucleic acid.
Yes. And what is the major difference in between them? Huh.
The major difference is the sugar in DNA is 2-deoxy D minus minus ribose, RNA D minus minus ribose, and DNA contains two alpha helix structure, two strands of alpha helix structure. RNA contains a single strand of alpha helix structure. DNA helps in hereditary transferring of characters from one generation to the next. RNA helps in the synthesis of protein. Then what else is there?
Enough. Good. Very good. Very good. Yes. Very good. Amazing. So yes, that is the major difference in between. The major difference in terms of its molecular basis is the sugar that makes it who it is, right? In the DNA, there is a deoxy sugar, and in the RNA, there is the ribose sugar, deoxyribose sugar in DNA and ribose sugar in RNA. Now see this. This is the picture of, not the picture, this is the chemical structure of a sugar, right? Ribose sugar because first carbon, second carbon, third, fourth, and fifth. 5 carbons are making this particular sugar. Okay. Now, at the second position of this sugar, if this oxygen is not present, now this is going to be called as deoxyribose. But when the oxygen is going to be there, we will call it as ribose sugar. Clear? So, this is a ribose sugar. And when we want to draw a deoxyribose sugar, so this is so carbon number 1 is over here, then it comes here. This is the carbon number 1 from it the OH group is attached. Here the fifth carbon CH2OH. Okay. Here again OH and here only H will. Okay. So here carbon number 1, carbon number 2, carbon number 3, carbon number 4 and carbon number 5. So here this one is deoxyribose sugar. This is the major difference in the structure of RNA and DNA. But here, if on the fifth carbon, okay, if on the fifth carbon, the phosphate group attaches and the nitrogenous base attaches to the first carbon, then we have a nucleotide. Clear? So if we
Yes.
talk about the nucleotide, the phosphate group is linked to the OH of the fifth carbon of the nucleoside. Through which bond? Through the phosphoester bond. Very important.
Okay.
Two nucleotides, also before that, the nitrogenous base is linked to the OH group, meaning the hydroxyl group of the first carbon of the pentose sugar, and that is through the N-glycosidic linkage. These bonds are very important to remember. So,
Could you repeat again the
see,
sentence?
the pentose sugar, first of all, we are going to make a nucleoside. So, to make a nucleoside, the nitrogenous base will be attached to the first carbon OH, meaning the hydroxyl group that is present near the first carbon of the sugar. So when this nitrogenous base attaches here, through which bond? This bond is the N-glycosidic bond. Okay, N-glycosidic linkage is there, which is going to make a nucleoside. And then at the fifth carbon, there will be the presence of the phosphate group attached by the phosphoester bond. And then we'll have a nucleotide that's made. So one nucleotide has two bonds in it, that is N-glycosidic linkage, and it has the phosphoester bond. Clear?
Yes.
So this is about one nucleotide. And then you simultaneously put two nucleotides together, then they will be linked to phosphodiester linkage in between them. Diester meaning phosphoester of the first nucleotide, phosphoester of second nucleotide, and they will be joined together. So phosphodiester linkage will be there, and the carbons attaching will be the 3' of one and the 5th carbon of another. So it will be sort of slant like that. Okay, the 5th carbon will be attached to the 3rd carbon of either of the nucleotides. Okay, and then when several nucleotides are present together, we call it as a polynucleotide chain. It starts forming a chain, like molecule after molecule after molecule, it starts forming a chain. Now, in a polynucleotide chain, the phosphate moiety, meaning the phosphate group, this is present at the 5' end because the phosphate group is at the 5th position of the carbon. And at the 3rd position, we have the OH group, which is the hydroxyl. Clear? So that is where the polarity of the DNA comes from, 5' and 3'. Why don't we say 2', 4'? This dash is also read as prime.
Once again. Yeah. So
What happened? What happened?
the fifth carbon, phosphate is attached.
Yes.
And at the third, next written here, and the third end of the sugar has the OH. Okay, okay, sorry. My bad. Yeah, got
5th
it.
of one, 3rd of another. Like
5th
that.
of
One.
like 2 nucleotides.
No, we are not talking about the 2 nucleotides. We have seen that several nucleotides are joined together. Now we're just talking about the ends. So one, the first carbon, or maybe if 50 nucleotides have joined together, so the 50th one. So the first one, the end one. So the first one will be having the 5' OH group, and the 3' end is going to have the OH group. The 5' is going to have the phosphate. Clear?
Okay.
And the backbone of the polynucleotide chain, it is formed by the sugar and phosphate. Backbone as in the core. The main thing that gives it a solid shape, solid structure, that is because of the sugar and the phosphate. Then what about the nitrogenous bases? They project from the backbone. So if this is the nucleotide, so the projecting from the backbone is the nitrogenous bases. Clear?
Okay.
But in the case of RNA, every nucleotide will be having one more OH group, which will be present at the 2nd carbon position, which was not there in deoxyribose. Why? Because in RNA, ribose sugar is there. Also, one more major difference is that in the case of RNA, like for example, if I talk about DNA, adenine pairs up with
adenine and guan. One second. Cytosine.
Adenine pairs up with
One second. Cytosine, guanine. One second, I can do this. Cytosine is guanine.
You only have one base left. What is that base?
No, there are 3 bases, right? Pyrimidine.
So which one pairs up with adenine normally?
I full ratta I did one second. Cytosine, guanine. Is it thymine?
Yes, thymine. Yes, adenine pairs up with thymine and guanine pairs up with cytosine. But this is what happens in DNA. But if you talk about RNA, so instead of the thymine, uracil is clear?
Yes.
I don't know why I mentioned this, but thymine's chemical name is 5-methyl uracil. It's irrelevant to the topic. But yeah, you should. Same way, different nitrogenous bases have different chemical names. Now, when we talk about the DNA, it is a long polymer of deoxyribonucleotides, right? Nucleotides are there, but deoxyribonucleotide meaning that it is made up of deoxy sugar.
Ma'am,
Now,
what is exactly ribon? Or I understood what is nucleotides. What is deoxyribo?
see, deoxyribo, this thing, for example, these two are the sugars, okay? Deoxyribose sugar, deoxyribo they write, ribo means ribose. Ribose means a sugar that has 5 carbons.
Okay.
Ribose is an example of a pentose sugar. Pentose means 5 carbons. Ribose is the name of the sugar that has 5 carbons.
Okay. Okay.
Got it?
Yeah.
Now,
Okay.
deoxyribose sugar is the ribose sugar that doesn't have OH group at the second carbon position. It doesn't have that OH group. Ribose sugar is the one that has normally the OH group at the second carbon position. Okay?
Okay. Okay.
Any doubts?
No.
So DNA, it is the long chain polymer of deoxyribose sugar or deoxyribonucleotide. And this was first identified by Frederick Mesker. He was a person who said that, okay, DNA is this, but he did not know it was called as DNA at that point. So he named it in his own language, nuclein. Okay, nuclein meaning suggesting that it is surrounded in the nucleus region. So nuclein. Okay. In the year 1953, James Watson and Francis Crick, now they started to study the DNA molecule and they wanted to deduce a structure of DNA. So they gave a double helix model for DNA to explain the structure of DNA. Like, why do we draw this DNA? Whenever you see a DNA being drawn, you see the two topsy-turvy lines, isn't it? Why? Because this is the double helical structure that we have been told that yes, DNA looks like this. Okay.
Ma'am, in every cell of our body we have DNA.
Yes. Everywhere.
Every cell.
Everywhere. Even in the tiniest parts of our body, we are who we are. It's because of the DNA.
Okay, in every cell there is DNA.
Every tiny structure has DNA. Even if like a drop of blood, even if a strand of hair, saliva, everything has DNA. Everything.
RNA.
Huh?
RNA.
RNA is also there in our body, but it's not our genetic material. That is why we couldn't extract RNA. But DNA is our genetic material. Genetic material means this is what we are made up of. RNA is also there in our body, but it has different purpose, protein formation, catalytic purposes, those kind of things. Okay?
Okay.
Now, Watson and Crick, they gave the double helical structure for DNA. But their research was based on the data that was produced by two people that were Maurice Wilkins and Rosalind Franklin. So they had already started to work with DNA and they did X-ray diffraction of it. Okay, X-ray diffraction is a method, it's like a experiment that they did and they produced a data. Okay, a set of data they produced. But they didn't know what to do with that data or what to deduce with the data. But then came Watson and Crick. They were new, they had better understanding of things. So they used or they sort of based their research on the data that was produced by Wilkins and Franklin and thereby gave the double helical model for structure of DNA. Okay?
Yes.
Any doubts?
No.
We're going to take this chapter slowly. So wherever you feel like the water is going above your head or the topics are jumping off of your head, you do tell. The moment any single thing jumps off your head, you have to inform me.
Okay.
Now, after that, there comes Erwin Chargaff's rule. Now, Erwin Chargaff, he gave a rule. Rule as in he said that, okay, yeah, one more thing. See, DNA and RNA, they can exist in two forms. Single-stranded, double-stranded. Okay, so mostly DNA that is present in every organism that is present as a double strand. Double strand means one helix, another together, yeah, twisted or intertwined. So according to him, in the double-stranded DNA, the purines and pyrimidines, they always occur in equal amounts, meaning however much purines will be there, that many pyrimidines. It will not be higher or lower. Okay.
Okay.
So the purines of the DNA are adenine and guanine, and the pyrimidines of the DNA are thymine and cytosine. And you already know that always a purine pairs up with a purine. Purine pairs up with a pyrimidine, right? Always.
Yes. Yes.
So adenine pairs up with thymine with two hydrogen bonds. These bonds, these are hydrogen bonds. Okay, remember that also. These are hydrogen bonds. So the adenine pairs up with the thymine with two hydrogen bonds, and the guanine pairs up with cytosine with three hydrogen bonds. And if you take the ratio, meaning the ratio of purines upon pyrimidines, the ratio will come out to be 1. 1 as in denoting the fact that it is constant for all species. It's like a constant value. It's an
What is it
integer.
exactly?
Eh?
Okay, ratio of A plus T plus C.
Meaning the ratio of purines to pyrimidines is constant for everyone. Meaning however much purines are there, that many pyrimidines.
Okay.
That's what happens. Like for example, once the screen is stuck. For example, 5 upon 5 is there, then you get 1
Okay.
this way. Right? If same numbers are there, they get cancelled out and the result is 1, which is a constant value. Now, the ratio is less than 1 in the case of eukaryotes, like in E. coli. What is the full form of E?
Equalite.
What is the full form of E in E. coli?
Electrolyte. Electro.
Electrocoli?
No, no, no. Equalite.
Hmm. Electronic coli. This is one of the most abundant bacteria there is. Most abundant.
Eubacteria.
Eh, Escherichia coli. You should know. Okay. I'll write it here.
What is it?
I'll write. My pen is not working. You write it Escherichia dot. I mean coli. This is the scientific name of E. coli, which is one of the most common bacteria there is. Okay. There may be some extra R in the middle, but this is what the name is. The ratio of, like in the case of eukaryotic organisms, like in humans, the ratio is more than 1, that is 1.5. Clear?
But aren't purines and pyrimidines equal in humans?
That's equal, but that's the ratio. Ratio meaning that it is like a whole number.
Okay.
It's not like anything less than, it's not in negatives, it's always positive. Clear? Any doubt?
Yes. No.
Okay, now note down. First, you do this thing. First, you write that genetic material is up to, you write the
Can I
heading
get
of the chapter.
can I get a PDF or something?
Huh?
Can I can I get a PDF of the notes?
You will be getting the PDF of all of the notes, but I still want you to write. Because, you know, I've taught you right now. It is fresh. It will sit in your mind like that. Okay? My intention is not to torture you, but to teach you. I will send you the notes of this thing, principle of inheritance, since that chapter is completed. Okay? Until then, you know them. It's okay, you can take your time. You don't have to be in a hurry. There is no marathon. But in my opinion, I feel like this is the ideal way to study because once you write, the thing gets imprinted in your mind.
Okay. Molecular basis. Yes, ma'am. Yes, ma'am. That nucleoside, the nitrogen space is linked to the OH of the first carbon of the pentose sugar during ribosome linkage. Yes, ma'am. Yes, ma'am. Yes, ma'am. Yes, ma'am. Yes, ma'am. Yes, ma'am. Yes, ma'am.
All right, any doubts?
No.
Okay, now let us see the various features of the double helical structure that Watson and Crick gave. So according to them, the DNA is made up of two polynucleotide chain. Okay. The backbone of the chain is made up with the sugar and the phosphate, and the two chains they run in anti-parallel fashion. Anti-parallel means 5 prime to 5 prime to 3 prime first chain and 3 prime to 5 prime second chain. This is the polarity of the DNA.
Prime? What is prime?
Prime means this dash, 5 dash.
Okay. It means the fifth carbon is attached to the third carbon of the other nucleotide.
Like fifth position, like which contains the phosphate group is present, then like that, like that, like that, and at the end there is the third position of the carbon that has the hydroxyl group present, like this. Same way, the anti-parallel chain to this will be the one that has 3 prime over here, and then several carbons later will have the 5 prime phosphate over here. Okay. And
Yes.
then the nitrogenous bases, they are paired up with the hydrogen bonds. Adenine is paired up with thymine, and guanine is paired up with cytosine by hydrogen, always. Now the two chains, they are coiled in a right-handed fashion, meaning their coiling is in this way, like this. Right-handed. Okay? Meaning the coiling is clockwise, not anti-clockwise. Always remember that. So right-handed coiling fashion is there. The pitch of the DNA. Pitch meaning the whole turn of the DNA. Like in the helix, these turns that are there, they are of 3.4 nanometer. And in every turn of the DNA helix, 10 base pairs are present. So one turn has 10 base pairs, another turn has 10 base pairs, like that.
A pair. A pair. 10 pairs.
10 base pairs. Adenine, guanine, cytosine, thymine, like that, 10 of them. Okay. Then the distances between like the base pair is 0.34 nanometer. It's not that the bases are like stuck onto each other. So the distances in between them would be 0.34 nanometer. Now, if you are given the number of base pairs and the distance between base pair, you already know. So by multiplying them, you can calculate the length of the DNA. The length of any DNA could be calculated if you are provided with the information of the total number of base pairs, and the distance between the base pair, you already know it is 0.34. So when you multiply these two values together, you will have the length of DNA. Clear?
Okay.
So this is what Watson and Crick said. Now there comes central dogma of molecular biology. This is like a phenomena that explains to us how the information flows from the DNA to the different biomolecules. Okay, so this was also given by Watson and Crick, and they said that there is a unidirectional flow of information from the master DNA to the copy DNA. Unidirectional flow, meaning the information flows in one direction and that is it. So this is what central dogma is. So from the DNA could be made, and that is called as replication. Okay, from the DNA, new DNA could be made, and we call that process as replication. And then the replication would undergo the process of transcription. The DNA would undergo the process of transcription and form the RNA. So RNA is made from the DNA by the process of transcription. Clear? And then this RNA will undergo the process of translation and form the protein ultimately. So from the DNA, information flows to the RNA, then from the RNA, information flows to the protein. This is the unidirectional flow, and we call it as the central dogma of molecular biology. Clear?
Okay. But what is transcription?
It is a process. RNA formation process is transcription. Protein formation process is translation. You have to study both. You have to study replication, translation, and transcription, all three. So now this central dogma, it is applicable on most of the organism except for one, which is viruses. So for that, there comes reverse central dogma. So this one is an exception to the central dogma of molecular biology. This was reported by Temin and Baltimore in 1970. That is why it is also called as Teminism. So according to them, viruses, there are some viruses, they have the enzyme reverse transcriptase. Okay. Using this enzyme, that virus could transform the RNA, like transform from the RNA a new DNA. So they can synthesize DNA from RNA. Imagine, from DNA, RNA was made in the right direction. But now from the RNA, DNA would be made in the opposite direction. So it's reverse of central dogma. See this. So from the DNA, RNA is made, but if you apply reverse transcriptase enzyme, from the RNA, DNA could be made. Now, this is an exception to the central dogma. Clear? Now, see, when we find out the number of base pairs that are present in human diploid cell, that is 6.6 into 10 to the power 9 base pair. And then when you multiply it with the number of, like the distance between the base pair, it comes out to be 2.2 meters. So the length of the DNA is 2.2 meters long of one cell. Have you seen like your mother's dupatta? Right? How long is it? Usually it's 2 meters dupatta. So imagine 2.2 meters, right? So the length of the DNA is 2.2 meters. Imagine that long. But our nucleus is 10 to the power minus 6 meter. Imagine how tiny our nucleus is, right? And the DNA is 2.2 meters. So how does the DNA fit inside the nucleus? Imagine, it has to be folded. It has to fit inside the nucleus, right? It has to be folded for that. And for that, we do the packaging of DNA. We don't do, our cell does the packaging of DNA. Okay. Why is it done? So as to package it, or so as to make it smaller to fit inside the nucleus. That's for eukaryotes. In prokaryotes also, DNA is, you can say, packaged, but it's lying there then like in the cell only. So in prokaryotes like E. coli, the DNA is scattered throughout the cell. Necessarily, there's no evident packaging that happens. Okay, and they always remember that DNA is negatively charged. It is a molecule that is negatively charged. So DNA is held with some proteins that are positively charged in the region that is called as nucleoid. So in the prokaryotic organism, since there is an absence of a nucleus, so the DNA is just going to be present lying there in the cell, and it will be surrounded, like DNA will be wrapped around or held with the proteins that are positive. So negative, positive, they attract each other, right? And that is how DNA is just present there in the cell of these organisms. But in the case of eukaryotes, the very complex packaging of DNA is done. Now here, the positively charged proteins on, like around which the DNA are wrapped is called as the histone proteins. Okay.
Last sentence.
Huh?
Repeat the sentence again.
The positively charged proteins around which the DNA is wrapped is called as the histone protein. Okay. They are the basic protein. Basic means they have OH group in them mostly. Okay. These are rich in the basic amino acids like lysine and arginine. Now these amino acids, they carry positive charge.
Inside histone, the DNA is wrapped.
No, see. No, not inside, outside. Have you seen top? Top as in lattu?
Yes.
The one, like how you take that top and you wrap the rassi or the rope around it and then you release it when it goes spinning, right? So imagine the wrapping. That's how DNA is wrapped around histone. So that top is the histone. The rope that you wrap around it is the DNA. Clear? Also remember one more thing, that proteins, they are made up of amino acids. All the proteins are made up of amino acids. So if a protein is made up of positive charge carrying amino acids, so that protein is a positive protein. And if it is made up of those amino acids that carry a negative charge, so they will be a negatively charged protein. But mostly proteins are positively charged. Okay. Now, first of all, what happens is that the histones, they get organized, like several histones come together, like 8 histones come together and they form a group. And that group or a unit of 8 molecules of histone, we, we call it as the histone octamer. Okay, 8 molecules are coming together forming the histone octamer. Now, the negatively charged DNA will start wrapping around the histone octamer, and then the wrapped DNA around the nucleosome will around the histone will be called as nucleosome. Clear? And a typical nucleosome, it contains 200 base pairs of the DNA helix. So where is the picture? Yeah, see this. These molecules are histones. Okay, several or eight number of histones, they came together and formed the histone octamer, around which the DNA is wrapped, and the structure is called as a nucleosome. Okay.
Okay.
From the nucleosome, chromatin is formed. What is chromatin? These are the thread-like beaded structures. It's like DNA, or you can say nucleosome arranged in the chromatid. It appears like a bead on string under the microscope. So histone, it forms the histone octamer, and when you add the DNA onto that, that becomes a nucleosome. From the nucleosome, it gets further coiled up, and it becomes the chromatin, and then the chromatin forms the chromosome. Clear? This is how
One second.
packaging of DNA is done. Huh.
One second. So histone is protein. So any proteins, histones come to, so histones are protein. So when they come together, 8 molecules, they become histones octamer. And around those, when DNA is wrapped around it, it forms nucleus. Again, further wrapping of DNA, further wrapping forms chromatin.
It becomes like entangled. Imagine like wrapping and wrapping and wrapping is done, and then it forms a bunch like a bunch. Okay. And that if you start organizing that bunch, then that will become a chromosome. Clear?
Yes.
So packaging of chromatin at higher level, it would require additional set of proteins that are called as non-histone chromosomal proteins.
One second. The packaging of
Even
chromatin.
more than that.
Packaging of chromatin at higher level requires additional set of protein. But it already forms chromosome, right?
That is true. But further, if let's just say there is more packaging that has to be done, so it would require higher level of protein. Okay. Now the chromatin, depending upon how they are arranged, it's of 2 types: euchromatin, heterochromatin. See, when the chromatin, they are loosely packed, okay, loosely arranged, we'll call it as euchromatin. And when they are tightly packed, we'll call it as heterochromatin. Okay, now see this picture. The first one, you see that all of them, they are tightly packed. The nucleosome along with the DNA, they are tightly packed, so we call it as heterochromatin. And the ones that are sparsely arranged or loosely packed, that is the U. Now, if something that is very compact, you try to stain them, they will pick up a darker stain. And if something is loosely like arranged, so they'll pick up a lightly stain, right? So the euchromatin region, it stains very light, whereas the heterochromatin region, it stains dark. But out of them, the euchromatin region is transcriptionally active, meaning this is the part of the chromatin that will undergo the process of transcription to form the RNA further on. Whereas the heterochromatin is transcriptionally inactive. Inactive meaning that it will not undergo the process of transcription to form the RNA further. Clear?
Okay.
After that, this is like the whole entire thing about the packaging. Just to summarize it all up. So you see DNA double helix is over here. It starts to wrap around the histones after the histones have formed an octamer. Then further wrapping takes place, which forms a chromatin. These materials, they form supercoils and they ultimately form a chromatin. And if you start arranging the chromatin, that becomes a chromosome. So this chromosome, huh?
Yeah, understood.
So a chromosome, always remember, a chromosome is made up of two chromatids. One chromatid, another chromatid, when it joins together at the center with the help of centromere, then you have a chromosome. Single unit of chromosome are called as chromatids. Clear? So there is a
Okay.
difference between chromatin and chromatid. You should be clear with that. Chromatin is the entangled mass of wrapped DNA. Entangled, it's like a bunch, tangled up. Chromosome is a more organized version of chromatin.
Yes.
Any doubts?
So this chromatin gets arranged, like the big thread chromatin gets arranged properly. Okay.
And then you can distinctly see the chromosomes. Okay, now you know down. Any doubts up until now? You've understood this topic, packaging of DNA. You understand why we need to package the DNA?
But why do we need to package?
You tell me. I already told you.
The packaging is done, everything is done, but then why? To give a lighter stain? I don't know. But why it is done?
Packaging is done because to fit the DNA in the nucleus. DNA is really long. How will you fit it without you? It's like, imagine you have a smaller space and you have to keep your clothes in it. So what do? So you shove it all in? No, you fold your clothes, put it one on top of the other, and then you put it inside the rack. Same way, when you have to fit the DNA inside the nucleus, you have to package it. You have to fold it so that it fits nice. Clear?
Okay, so it's all is done just to fit the DNA into the nucleus.
Yeah. Or how else will you fit a particular DNA inside a nucleus? Because DNA is 2.2 meters long. Imagine 2.2 meters is the length of a dupatta that we wear, right? And nucleus is 10 to the power minus 6 meter. 10 to the power minus 6 means
Yeah, very less.
like this. So what would it be? 0.000001, like that, isn't it? That's going to be the size of the nucleus. So that's how tiny the nucleus is. So in order to fit it, you have to fold it. Clear? That is why we do the packaging of the DNA so as to fit it inside. Got it?
Yes, ma'am.
Huh. Now you know it.
Yes, ma'am. Yes, ma'am. Yes, ma'am. Yes, ma'am. Yes, ma'am. Yes, ma'am. Ma'am.
All right. Any doubts up until now, Sanjita?
Ma'am.
Okay. So let's just keep it till here only, today's class, and we'll continue in the next class. Okay.
Okay.
All right.
Thank you.